Assalamualaikum, my name is Farhah and along with me is Sofia Dinie. We will be presenting a poster presentation on our ERP project entitled Effects of Probiotic-Containing Functional Foods on Dental Enamel Structure, a Systematic Review. I will start with the introduction, methods and results. On the other hand, my friend Sofia will present the discussion, strengths, limitations, and our conclusion that we came across. Ladies and gentlemen, allow me to introduce the scope and aim of our review. As we all know, dental caries is a worldwide oral health problem which is defined simply as a localized breakdown of vulnerable dental health tissues. There are many factors that contribute to dental caries, including host, substrate, bacteria, and time. In order to combat dental caries, WHO has recommended several practices in order to help prevent dental caries, such as the use of fluoride, better oral hygiene procedures, and proper dietary practices. Focusing on dietary practices, functional food worldwide plays an important role in promoting oral health, as their aim is to provide additional benefits beyond basic nutrition. Functional food is defined as enriched food that have a positive impact on the human body. Currently, functional food market is dominated by symbiotic, prebiotic, probiotics, fatty acids, dietary fibers, carotenoids, vitamins, and minerals. Functional dairy products account for more than 40% of the functional food market. Probiotic-enhanced dairy product is a subset of the functional food domain and is what this review is focused on. Probiotics is defined as a live microbiome food supplement that when taken in adequate amount aims to exert health benefits on the host. Lactobacillus and Bifidobacterium are the most common genera utilized as strains in these food products, as they are mainly used for their proven gastrointestinal health benefits. However, their documented benefits toward oral health is limited. This in an attempt to understand the role of probiotic-containing functional food in oral health, specifically their role in influencing enamel structure. The aim of this evidence-based analysis is to look at the original research papers that investigate the effects of commercially available probiotic-containing functional food on dental enamel structure. Moving to the methodology, in order to do this, we choose a set of keywords that our literature search will be based on. We choose keyword number 1, probiotic, number 2, enamel, or demineralization or remineralization. As we wanted to look at recent studies, we narrowed our findings to the last 2 decades, starting from the year 2000 until 2020. We chose 2 databases, Mazlan and Syst, as we wanted the studies to be health-related. The main inclusion criteria were number 1, published in English. Second, studies use commercially available probiotic-containing functional food. And number 3, studies had to report on the association of probiotic-containing functional food on dental enamel structure. Our exclusion criteria were studies that number 1, isolating a specific probiotic strain and not studying the effects on enamel structure. Number 2, reviews, news, letters, editorials, and case studies. And number 3, animal studies. This flowchart shows the steps we conducted in order to eliminate irrelevant articles. As for the results, our initial search gives us 642 studies from both databases. After going through the articles based on the inclusion and exclusion criteria, excluding duplicates, we were left with 9 articles. We then tabulate the key findings from the 9 articles as shown in Table 1 here. So, our review consists of 3 studies using bovine dental enamel blocks, while 6 studies using human permanent or deciduous teeth. 2 of the studies using human teeth were conducted in vivo, while 4 studies were conducted in vitro. Out of 9 studies, only 1 study did not use dairy products as the mode for probiotic delivery. However, the other 8 studies used commercially available dairy-based probiotic products such as milk and yogurt as their functional food of choice.
Thank you, Alya, for explaining regarding our introduction, methods and result. As the aim for our review, we wanted to look into the effect of commercially available probiotic functional foods on dental enamel. We specifically focus on the enamel structural changes, which is mineral loss. Overall, from the selected 9 articles, what we found were mixed findings. There were both positive and also negative findings with regards to the enamel structural changes. There was a strong evidence of benefits of the probiotic functional foods on enamel, as 56% of the reviewed article has positive changes in minimizing the mineral loss on dental enamel. Lodi et al. In his study found that treatment with Bata-V2 fermented milk had a significant lower percentage change in enamel surface microhardness in comparison with treatment with Yakult and control group. The Bata-V2, a fermented milk, contains 3 probiotic bacteria, has seen to yield better than Yakult, which only contained a single strain probiotic. This is because application of different probiotic bacteria affects the balance of the oral ecosystem via possible additive, cumulative, and competitive modes of action. However, there are 2 papers, Alya et al. And Shan et al., which reported negative findings, while 2 papers reported no significant effects. According to Alya et al., probiotic-containing milk formula had the highest degree of dimerization due to the presence of 2 bacterial species in carriage development through multiple mechanisms such as acid production and resistant to acidic exposure. The 2 lactic acid presented in the formula milk was Bifidobacterium and Lactobacillus. One of the most important characteristics leading to their carcinogenicity was the acid byproduct of carbohydrate fermentation. Shan et al., on the other hand, discovered that commercial yogurt containing live probiotic culture further reduced the enamel mineral content by 320% than the initial lesion due to the presence of added sugar in the drink, which will later promote caries to take place. The yogurt contains very high lactic acid concentration, which inhibits the buffering of the artificial saliva and created an acidic environment. Based on the issues above, the main issue regarding the effects of commercial probiotic drinks towards human body is that it is a double-edged sword, as you can see from the bottom left poster. Even though the probiotic have a positive effect and provide beneficial bacteria for the gastrointestinal environment and can prevent and compete with harmful oral microflora, there are 2 factors that can negate this therapeutic effect. The 2 factors are the high sugar content in the beverages itself and the acidic nature of the probiotic bacteria. According to our literature search, this is the first systematic review on commercially available functional food products and their impact on dental enamel structural changes. Our analysis included the most recent published article in our area of focus, covered both in vivo and in vitro study, as well as providing a hypothesis on why we saw mixed findings regarding this matter. This study has limitation. Several studies fail to distinguish the probiotic bacteria strain used in each of the probiotic functional food products studied. Other criteria such as pH, bowel film, blood count were also included in most and many of the articles, thus making the analysis on its effect on dental enamel was not standardized among all the studies. Apart from that, several studies were conducted using the bovine dental enamel blocks. In conclusion, further studies are needed to determine the ideal properties of probiotic functional food products, such as having low capacity of acid production. The probiotic strain with antimicrobial property, pH buffering effect, and less acidogenic would be ideal for the general public. That's all from us. Thank you.